Okay, on deck, we have oral presentation number 16. This is Downingtown, Pennsylvania. This is a Mission 4 flight experiment. So Mission 4 has returned to Earth back in March.
Today we will be presenting about microencapsulation in microgravity. We're from Downingtown STEM Academy. My name is Avni Glorjani, and joining me in this presentation are Miranda McMillan, Christina Murray, Jenny Faust, Cecilia Padilla, and Santino Zoras. Megan Joyce and Connor McGrath are also part of this experiment, but were unable to make it today, unfortunately.
So our experiment examines the function of microencapsulation in microgravity, and basically microencapsulation is a common process used to create longer-lasting medications by coating the active ingredient of the drug in an inactive polymer. Now, most medications are microencapsulated because it allows the drug, or prevents it from being released too rapidly within the body, and this decreases the frequency that a person must take a drug, and it also makes it much safer to take a medication. So in our experiment, we examine the concentration of naproxen, or the active ingredient in the medication Aleve, which induces pain relief. And by examining the concentration of this active ingredient, we are able to see whether this microencapsulation, or this inactive coating, still creates this controlled release in microgravity. Next.
So we're testing the concentration of naproxen because it is the ingredient in Aleve that is biologically active. Therefore, investigating the amount of naproxen will allow us to determine the effectiveness of microencapsulation in microgravity. If the concentration of naproxen is less in the space experiment compared to the ground experiment, this indicates that the microencapsulation was stronger. However, if there is less naproxen, if there is more naproxen that is dissolved in the space experiment compared to the ground experiment, this indicates that the microencapsulation was weaker because more naproxen was able to be dissolved.
Okay, next slide. So for our experiment, we chose to use the Type 3 FME mini lab. So that was the mini lab that was able to hold 3 different volumes. And in our mini lab, the first volume, volume 1, was Aleve extended release microencapsulated medication, represented by the blue pill. And in Aleve, naproxen is the active ingredient, and naproxen is what's microencapsulated. So as Christina and Jenny just said, this simply just means that it has many coatings that dissolve at different rates. And then our central volume was a simulated stomach acid, and our last volume in the tube was an antacid, Mylanta. So basically, for our experiment, what we had them do in space is we had them first remove the first clamp between volumes 1 and 2, the pill and the acid, and this allowed the acid to dissolve the pill. And then 2 days later, we had them release the second clamp, and this allowed the antacid, Mylanta, to essentially neutralize and stop the reaction once it mixed with the remaining solution. And then from there, upon returning home, we were able to take the solution from our tube and measure the amount of Aleve XR that had dissolved, and we used a special technique to do this.
Oh, next.
Yeah, and next slide again. Sorry.
To analyze the solution and find our results, we used a method called thin layer chromatography, or TLC. TLC separates different molecules in a solution and shows its concentration. To perform thin layer chromatography, a tiny portion of the solution being tested was put on a TLC plate. The TLC plates were placed in a shallow pool of solvent and left to run until the solvent reached the top of the card. The solvent travels up the card via capillary action and takes the solution being tested with it. After the cards are done running, they're taken out and placed over a UV light. The UV light reveals the spots of the solution, and results can be drawn from these spots.
In order to find a standard curve that could be used to determine the exact concentration of naproxen in the space and ground sample, 4 samples of unfiltered ground tests were spotted in 7 TLC plates with different volumes by using a micropipette. The samples that were going to be compared, the space and earth one, were filtered, and then 3 microliters of each was spotted on each of the plates. The spotted TLC plates were then placed in individual beakers, each containing isopropanol, which acted as the solvent that essentially separated the naproxen from the samples. The test was run, and then the TLC plates were taken out and dried. The dry TLC plates were then placed on top of a UV light box to determine the amount of naproxen in the samples by looking at the intensity of the colors. Basically, the darker the color, the more concentrated the sample is. We then analyzed we took pictures and then we analyzed them by using an ImageJ program.
So the concentration of naproxen was found by weighing the peaks of the graph that was found through the ImageJ program. The concentrations were then plotted on a graph that gave us a standard curve. And a standard curve is basically a graph that allows you to determine the properties of unknown samples through the interpolation of the graph that already has plotted the points of samples with known properties. And through this standard curve, we were then able to derive a linear equation that would allow us to calculate the concentrations of naproxen in both the space and ground experiments.
Next slide. So here you can see there's the intensity curve. So once that was found, once we derived this equation, we were able to see that plug in for the y value and eventually find for the x the unknown concentration of naproxen. And we took an average of these 7 trials. Next slide. And from these trials, we were able to see that more naproxen dissolved in solution. And this told us that microencapsulation in microgravity is less effective because more naproxen dissolved.
We later performed our second experiment to verify the previous results because the previous experiment showed a saturative curve, meaning that the maximum amount of naproxen in the pill had been dissolved. So this time we spotted 4 additional TLC plates, but this time with smaller samples. And what we concluded was that the difference in the concentration of naproxen in the space and ground test was not drastic enough to have any sort of damaging effect on the user.
This experiment is important because microencapsulation used in many common medications to treat many common ailments. With commercial spaceflight becoming a reality, it is good that we tested this method of making medication that's very common.
Thank you very much. Are there any questions for the Downingtown team?
Michael from Downingtown. So throughout this course, it sounds like the experiment in space was less effective. Was that surprising to you? What were your expectations?
We actually expected it to actually be stronger because they tested the making of microencapsulation in space, and the crystals kind of that make the microcapsule are actually stronger in space. So we wanted to test whether if you put the medicine already microencapsulated, whether it would work up there. So from those previous tests, we thought, maybe it would be stronger, but it was surprising to see that it was less effective. And so that was something that we kind of had to reflect on and see what the difference was.
Tim, Goddard Space Flight Center. So I thought the results are very interesting. There was something that intrigued me. I was looking at the thin plate chromatography. Would the method that you used there, would that work in microgravity, or does that depend on gravity to make it effective?
Well, the components would travel up using capillary action, so I don't think that thin layer chromatography would work in space because there's no gravity.
Yeah, I also don't think, because it has to sit in a shallow pool of liquid because liquids act differently in microgravity. I don't know if that would be as possible, so it might not even work because it might not even be able to sit in a pool of water.
Okay, thank you.
Okay, let's thank the Downingtown team. Superb job.